Welcome to 5-Minute Papers on AI for the Planet. I'm Grace Lindsay. Our paper today is Barcode Mamba, State Space Models for Biodiversity Analysis. Let's get started. DNA contains the instructions for life. It encodes how an organism looks, grows, and acts. And the fact that a copy of these instructions is stored inside every cell is really helpful for scientists interested in studying biodiversity. Out in the field, scientists can collect DNA samples from animals directly, or even from the cells they leave behind, like in shed feathers or scales. From this, they can extract a small region of DNA called a barcode and use it to identify the species without having to visually inspect or observe it at all. This technique of DNA barcoding allows for more accurate biodiversity analysis in the wild at scale. However, mapping these barcodes, which are hundreds of base pairs long, to a species classification isn't a completely solved problem, and there are still plenty of species whose barcodes have never been analyzed at all. To help make the process of barcoding faster and more accurate, this paper builds a foundation model for barcode-based species classification. Past work to tackle this problem has mainly used feed-forward model architectures, such as convolutional neural networks and transformers. But this paper builds on recent advances in state space models, which are a form of recurrent network, wherein representations are updated over time steps. Specifically, this paper uses the Mamba architecture. Each input in a Mamba network is fed into a state space model, where it is used to update the current hidden state. The recurrent weights, labeled here as A, determine how information from past inputs combine with information from the current input. To allow for more complex interactions, the matrices that map inputs to the hidden state and that map the hidden state to the output, labeled here as B and C, are allowed to change on each time step based on what the current input is. This compact way of blending information across inputs provides a more computationally efficient alternative to other sequence models like transformers. Here, the authors use Mamba out of a belief that its structure will also be better able to handle variations in DNA sampling length and quality. To train their Mamba model, the authors use data from the Barcode of Life data system. Specifically, they use the Canadian invertebrate dataset, which contains 1.5 million samples of invertebrate DNA. Classifying invertebrate species can be particularly difficult due to the large number of species, their complex relationships, and the large imbalances in available data. Each sample in this dataset contains a DNA sequence and the corresponding species label. The DNA sequence is stored as a list composed of 4 possible characters, A, T, G, or C, which represent the nucleotide base pairs that make up DNA. The character N is also used to indicate missing or uncertain data. In this work, each DNA sequence is cut to be 660 characters long. The goal of the model is ultimately to take in a sequence of characters and output a correct classification for the animal. To find the best possible model to do this, the authors explored many different variants of Mamba. First, they had to decide how they would feed the DNA data into the model. One option is to feed it in one character at a time, which would result in a vocabulary size of 5 and a sequence length of 660. Alternatively, the authors tried chunking characters into groups of K. This increases the vocabulary size while decreasing the sequence length. Next, they explored different ways of pre-training the model to allow it to learn good representations. The first method, which is commonly used to train large language models, is next token prediction, where the goal of the model is to predict what comes next in a sequence. The authors also tried masked language modeling, where the model needs to fill in a missing input in a sequence, but found it performed worse. Finally, the authors explored varying the number and size of the layers in their model architecture. Through these tests, they settled on 2 versions of Barcode Mamba to explore, a mini one with up to 7.4 million parameters and a large one with 56.7 million parameters, which took over 42 hours to train. To test if their architecture and pre-training lead to useful representations, the authors explored Barcode Mamba's performance on species classification tasks and compared it to several pre-existing genetic models, including those with transformer-based architectures, convolutional neural networks, and other state space models. In the first test of performance, the models are fine-tuned to perform species classification. Given that in fine-tuning, model weights are allowed to update to improve performance, all models do well on this classification task. The authors then use linear probing as a stricter test of model representations. Here, only the readout weights that map hidden layer activity to species labels are allowed to update. In this case, Barcode Mamba's performance exceeds that of other models. Finally, the authors explore how well the models can predict an animal's genus without training any weights at all. Specifically, they apply a nearest neighbor approach to the model embeddings to make an informed guess about the genus of a previously unseen species. Here, Barcode Mamba significantly outperforms other models, and it does so with far fewer parameters. In total, the authors show how the Mamba architecture can be effectively used to create models that turn DNA sequences into genus and species classification. In the future, they hope to test the model's performance on the newly released BioScan 5M, which contains 5 million insect DNA samples. That's all for this episode. Remember to subscribe to this channel for more examples of how AI can be used to understand and protect our planet.